Hello again and welcome to another video. In this video I'm going to talk about the mechanism of cellular immunity, which is one of the four main mechanisms we have in our body. Now we already looked at humoral immune response and immunological memory. Now let's go ahead and look at what cellular immune response is. So this is what you need to remember in terms of cellular immune response. It's that cellular immune response has something to do with killing other cells. Now which cells are their target? First one is obviously going to be cells that have been infected with an intracellular microorganism. Also could be oncogenic cells or cancer cells. And also can be, transplanted cells that are not initially yours. Now, why are all of these three the target of your cellular immune response? The answer is the MHCI. You can think of MHCI as kind of it provides you a status update of the cell, and if the peptide that the cells present on the MHCI is not recognized as normal, it'll be killed. So a virus infected cell puts a fragment of the virus on the MHCI, so it's killed. An oncogenic cell puts a fragment of the mutated amino acid, for example, on the MHCI, and that is not normal, and then that's why it's killed. And then a transplanted cell of the other person's normal peptide is usually foreign for the person receiving the organ. Now, what cells are responsible to clean up the body from these cells? First one's obviously going to be T-Cytotoxic Lymphocytes. Natural killer cells are like the warriors of cellular immune response. Then we got macrophages. These guys are everywhere. So let's go through the mechanism of each of these to see how they work in the cellular immune response. We can start with the T-cytotoxic lymphocyte. These guys are the kind of T cells that go hunt after a sick cell once they're activated. And just like any other T cells, they need three activation signals to activate. So the first activation signal is what we call T cell receptor. Notice that it has CD8 instead of CD4. CD8 is going to dock on MHC1 molecule. And if you usually forget which MHC binds to CD4 or CD8, the way I remember it is that I always take the highest with the lowest number. So CD4 is the lowest, goes with the highest number of MHC, which is type 2. CD8 is the highest number of CD4 and CD8. So it goes with the lowest MHC type. So CD8 with MHC1 and CD4 with MHC2. So anyways, T cell receptor with CD8, and it has also CD3 for inducing a signal to the cell after it binds. So that is the first signal. It also expresses CD28 so that it can bind to B7 for the second activation signal. The third activation signal involves receiving interferon gamma and interleukin 2 from an active T helper cell 1. And that's why T helper cell 1 has something to do with a cytotoxicity as well, not just activating macrophages and activating inflammatory response. Th2, however, has more to do with B cell activation to get antibodies, so they're more over to the humoral immune response. So T helper cell 1 helps cellular immune response. When it receives those signals, it can finally proliferate and differentiate into effector T cytotoxic cells. So once T cells has been activated, they start expressing FAS ligand. What do is that, when cells are infected, they usually express FAS, so that when the T cytotoxic cell binds, it's going to kill it by releasing perforins, which perforates the membrane, and then granzymes, which fragments their DNA. So that was mainly how the T cells work. I'll show you a scenario later on how this goes. But now let's look at natural killer cells. These cells, so they're like the policemen of your body. They're also able to recognize unhealthy from healthy cells. And actually the fact that they're able to do that by their surface receptor, which they already have on their surface, makes them a very important part of the innate or natural immunity, not the adaptive immunity. Alright, keep that in mind. The natural killer cells are a part of your innate immunity because these guys are already able to recognize sick from healthy cells. And I'll show you how. On the surface of the natural killer cell, you're going to have something called LY49. And, normally cells express MHC1, right? All nucleated cells in your body express MHC1. And the LY49 is going to bind to it And once it binds to the MHCI, it's going to induce a negative signal to the natural killer cell so that the natural killer cell doesn't kill it. Another surface receptor it has is something called NKRP1 or natural killer receptor P1. So, when the cell is virus infected and it puts a little fragment of the virus on the surface of the MHC1, the thing is, natural killer cell cannot recognize the peptide like a T cell receptor can, but all the cells in your body expresses a molecule on the surface which can be recognized by the natural killer receptor P1, and then that sends a positive signal to the cell to kill the cell. Now keep in mind that these molecules on the surface of the cells that bind to the natural killer cell receptor P1 are usually always present on the surface of your cell, but once a cell is infected, it tends to reduce the amount of MHC class 1 molecules on its surface, which means less Ly49 can bind, and then again less negative signals to the cell, so natural killer receptor P1 takes over. So that's mainly how the natural killer cell can differentiate between healthy and sick cells. It kills depending on how many MHC class 1 molecules a cell has on the surface. And so now what happens? The natural killer cell is going to release some granzymes which is going to kill the cell respectively. Another receptor that the natural killer cell is going to have is the Fas ligand. Remember that one? The same one as an effector T-Cytotoxic cell. Remember, when a normal cell gets infected with an intracellular microorganism, it's going to express FAS. Natural killer cells can recognize the FAS by the FAS ligand, and then it's going to release perforins, which penetrates the membrane, and then granzymes, which fragments the DNA to then kill the cell. We also have something called CD16. So, in a normal cell, when it gets infected with a virus, more specifically, when it gets infected with a complex virus, usually what happens is that the viral nucleic acid becomes integrated into the genome of the host cell, right? What can happen is that, complex viruses gets out from the cell through budding, right? It just kind of fuses with the membrane and just goes out. Usually, as it prepares to go out through budding, it expresses some parts of the viral peptides on the surface of the cell. During that time, an antibody might bind to the surface of the cell. And what happens then, CD16 is going to bind the Fc region of the antibody. And CD16 is actually, it binds specifically to the Fc region of an IgG type antibody. Alright, so this type of antibody right here is an IgG. Alright, and then after that, it performs what is called an antibody-dependent cytotoxicity to kill the cell. Alright, so now we got Ly49, natural killer receptor P1, FAS ligand, and CD16. It also expresses interleukin 2 receptor, which binds interleukin 2 and gets activated. Remember, I just showed you that the T helper cell 1 helps the T cytotoxic cell activate by releasing interleukin 2 and interferon gamma. Here you can see that T helper cell can also activate natural killer cell by the help of interleukin 2. Other things it does is that an active natural killer cell releases tumor necrotic factor alpha. We've already talked so much about this one every time we mention the macrophage, because this cytokine is able to help with the inflammation process by, activating endotheliocytes, by releasing antimicrobial proteins, and also expressing receptors to help cells get to the inflammation area. Tumor necrotic factor has also a systemic function that it causes fever by going to the hypothalamus of the brain to release prostaglandin E2 and cause fever. The natural killer cell can also release interferon gamma. I'll talk more about the interferons at the end of this video, but interferon gamma has a lot of functions. It can help cytotoxicity by activating more T helper cell 1. It can activate other natural killer cells and also macrophages, and it can also stimulate the B cells into releasing IgG. So that's those. It also has an MHCI. It's kind of logical because remember, MHCI is present on all nucleated cells, and natural killer cell also has nucleus, so it also has MHC1. Natural killer cells also have MHC type 2 molecules. It's not really proven why they have MHC class 2 molecules because they're not antigen presenting cells. That's why I haven't included it, but if you read somewhere that they have MHC type 2, it might be true, I'm not sure. And so another thing that's really important is that it has the surface markers CD56 and CD16 and also has CD2, which helps with adhesion. CD56 and CD16 are markers, not CD2. So that was everything I had for the natural killer cells at least. Now let's play a scenario. Let's say this virus just infected the body. It's either going to infect any type of cells you have in your body, or it's going to directly get eaten by an antigen presenting cell. When a normal cell gets infected in your body, it may present a part of the virus on an MHC type 1, becoming what we call non-professional antigen presenting cell, as it lacks B7. That's why it's non-professional. Remember, professional antigen presenting cells show B7. So if it infects a normal cell, the antigen presenting cell can phagocyte the whole cell. Either way, it's called a professional antigen presenting cell because it can express the important core stimulator receptor B7, as well as presenting the peptide on an MHC class 1 or class 2. Now remember, a T-cytotoxic cell has TCR receptor, which include the CD8, which binds to MHC class 1, and CD3, which induces an internal signal once it has bound. This is the first activation signal of the T-cytotoxic cell. The second signal is B7 binding to CD28. The third activation signal comes from a T helper cell 1. So we need this professional antigen presenting cell to kind of cross-present this peptide, this virus. And a dendritic cell is really, really good at cross-presenting because this cell is really big and it has dendrites which can extend to a lot of cells and activate many cells at the same time. So while it's trying to activate the T-cytotoxic cell, it's also trying to activate the naive T helper cell. And it does that through the T cell receptor MHC2 and through the B7 CD28. And it also are able to differentiate it into a T helper cell 1 by releasing interleukin 12. It can also receive interferon gamma from natural killer cell to get differentiated. Now that the cell is activated after receiving the cytokines, it needs to grow to be able to divide, right? And it does that through releasing interleukin 2, which sits on its own surface. The process of releasing cytokines, which sits on your own surface, that process is called autocrine. So the interleukin 2 has an autocrine function. So now it becomes a T helper cell 1, and the T helper cell 1 can give the third activation signal to the T-cytotoxic cell, which is the interferon gamma and interleukin 2, so that this cell can differentiate and proliferate to an active T-cytotoxic cell, which will express FAS ligand. Now, remember I told you that the virus-infected cells express FAS receptor, right? As you see right here. And after the T-cytotoxic cells has bound to it, it's going to release perforins, which perforate the membrane, and it's also going to release granzyme B, which fragments the DNA and then kills the cell. But now we only have some free-floating viruses. You know the T helper cell 1? It has another really important function of expressing CD40 ligand, which can bind to CD40 on the macrophage. And then together with CD40 ligand and interferon gamma, it activates the macrophage into engulfing the remaining viruses, the free-floating viruses. Now, immunity to viruses can happen in two ways, really, even adaptive or innate. All of this I just showed you involves T cells, and when T cells are involved, then we shift automatically to adaptive immune response. Because remember, an adaptive immune response gives an immunological memory of these viruses and also produce antibodies against it since T cells are involved. And I'll talk about how antibodies react to viruses later in this video, but the other main type of immunity against viruses is going to be what we call innate immune factors. Now, there are three factors of the innate immunity that gives off a non-specific defense. The first one is general factors. Viruses attacks host cells via specific receptors, right? And this partially determines which cell types becomes infected. This is called tropism, when the virus has specific receptors on a host cell that it attacks. And if the host cell doesn't really have those receptors, the virus can't really infect it. That is what we call tropism. We got neurotropic viruses, we get enterotropic viruses. So all of those are very specific to what kind of cells they target. So this factor is an example of general factors we have for the innate immune response against viruses. Another way is, macrophages? It can actually release inflammatory cytokines once activated, and they will go to the hypothalamus of the brain and cause fever reaction. That when the temperature increases, it can make harsher environment for viruses and certain type of bacteria as well to live in. So that's also a general factor of the innate immune response against viruses, fever. It could also be secretory functions by feces, for example, or saliva. Those are also general factors. The second type of innate immune factors against viruses are humoral factors. The complement system are all the time released by the liver. The complement system can also opsonize the bacteria and help fight against it, as well as interleukin 6 can also go to the liver to cause it to release acute phase proteins like C-reactive proteins or mannose-binding lectin. The mannose-binding lectin protein plays an important role in humoral defense especially against the influenza virus infection, for example. Keep in mind that the mannose-binding lectin is a part of your complement system. The last type is called cellular factors, and one of them is phagocytosis. Phagocytosis can happen when the phagocyte just binds to the virus, or even it can happen indirectly if the complement system opsonizes it, or it can happen directly if an antibody binds to it. But another type of cellular factors are what we call interferons. So let's look at how these work. The interferons play a crucial factor in defense against viruses, and they are very important signal proteins released by host cells, often in response to several viruses. So there are also cytokines, and there are many interferon types in our body, but I'll mostly talk about these three types: interferon alpha, beta, and gamma. Interferon alpha is also called leukocyte interferon because leukocytes releases them. And interferon beta is the fibroblast interferon because fibroblasts release them. Interferon gamma is the immune interferon released by several immune cells. And we usually group these interferons though according to the type of receptors through which they bind. Interferon alpha and beta are type 1 interferons because they bind to specific receptors called interferon alpha beta receptor. Interferon gamma are type 2 interferons, usually activated by interleukin 12. So let's look at type 1 first and then look at what type 2 is. So as the cell gets infected by the virus, the genes that produce interferon alpha and beta are located on chromosome 9, right? But usually on normal cells, these genes are blocked. But these infected cells, the virus that infected them triggers interferon inducers to remove this block right here completely. And when you remove this block, it starts to produce interferon alpha and beta. So what do they do? Well, for one thing, once the block is gone, interferon alpha and beta will inhibit the protein synthesis and the DNA synthesis in the virus infected cell to stop the virus from replicating. Then it will increase the MHCI type 1 expression on all the cells to protect them from this mess. They will also promote and activate natural killer cell to kill it. Interferon alpha and beta will also activate or actually stimulate T and B lymphocytes, so they can also help to kill this virus and other infected cells. So that is mostly type 1 interferon. Type 2 interferon, however, remember they're interferon gamma, and they could be released by either T helper cell 1, T-Cytotoxic cell, and macrophages. So when an effector T helper cell 1 releases interferon gamma, is going to reduce the amount of T helper cell 2 we're going to have in the body. Why is that? It's because the moment the viral infection happens in your body, you need more T helper cell 1 than T helper cell 2. So T helper cell 2 is switched off and T helper cell 1 production increases. They also activate macrophages and natural killer cells and also stimulate B cells into differentiating to an IgG producing plasma cell. All of these is what the interferon gamma does. It's released also by an active natural killer cell and also an active T-Cytotoxic cell. Now you see these IgG immunoglobulins. Remember I told you that the adaptive immunity against viruses also cause antibody to get released? These are produced by plasma cells. And in the first week of infection, IgM is going to be circulated around your bloodstream to neutralize the circulating virus. And remember the humoral immune response when I told you that IgM is going to be the fastest produced antibodies because it's produced without the help of T cells. But then during the second week, after all the T cells have reacted and B cells reacted and all that, IgG is going to be released. So that's why IgG takes a little more time. And they are going to circulate around your body and look for viruses. And they can also catch extravascular viruses, even when the viruses are inside the cell. As a result, you can also find IgA on the mucosal surface to protect against reinfection. So what type of acquired immunity do we have against viruses? There are a lifelong immunity, like measles for example. We also have low-level immunity. Those are usually chronic viral infections. Or we can have no immune response. Those are slow viral infections like prions, for example. Those are just proteins. Now, let's sum up. A virus-infected cell can show FAS for T-cytotoxic to kill it by releasing perforins and granzyme B. Another way is by T helper cell 1 activates macrophage with the help of CD40L and interferon gamma, which will then phagocyte the virus. Natural killer cell can kill it by either surface receptors, and even antibodies can opsonize the cell and cause other cells to bind to the antibody and kill it. Or even it may activate the classical pathway of the complement system. Another way is that if you have a free-floating virus, antibodies can also opsonize it as well. So that was everything I had for the cellular immunity. Lastly, let's look at how hypersensitivity goes.